Hi, good afternoon, everyone. So, I'm Surabhi, and I'm a PhD student at IIC Bangalore currently. And uh, for this semester, for Basics of Biology course, I'm one of the TAs where I will be taking tutorial classes, and and we'll just brush up the week by week content, and then look at few questions. All right. So, because we have actually a lot of people, approximately 28 people, uh, in this call now. So, if quickly everyone can just maybe write in the chat of like about where are you from and like what is your background currently, like you're doing BSc, MSc, BTech, MTech, or whether you're in school or high school, something. It would be good to get an idea of what is the diversity of the audience that we are going to address. Okay, feel free to write in the chat. Hi Neha. All right. Or while someone's writing in the chat, people can raise their hands and unmute themselves to introduce unmute and like via audio. And if people are okay writing in the chat, you can go ahead and write. So, hi Mrinalini. Hi Nishat. Hi Priyankshi. Hi Sana. Yeah, Monisha, you can go ahead. Hi Geetika. So meanwhile, while people are introducing themselves, I will start as well. Hi Devesh. People are waiting to join. People are joining still. Hi Darshini. Hi Kavya. All right. So what I'm seeing is mostly people are from like people are doing BSc. They are in their first year. Hi Ashika. If you want to go ahead and unmute yourself and speak, you are welcome to do so. Ashika, you want to go ahead and say something? All right. So maybe it's 3:12 now. Hi Kirtana. All right. So I will start with today's session. Yeah, sure, sure. Hi, hi Neha. So currently I'm a fourth year PhD student at IIC Bangalore, and what I work on is at the interface of protein biology, where we look at the structures of translation complexes. Uh, like in our lab, we use like biochemistry and molecular biology approaches, as well as basically we predominantly do cryo-electron microscopy to look at the structures of translation initiation complexes in plants, bacteria, fungi, uh, and that's how we are. Like different students are doing so to address, to look at the fundamental aspect of translation, protein synthesis. Hi, Maria. So, hi Koil. Actually, I did my integrated masters in like BSMS from Izar Barhampur. It was like I didn't have any particular subject per se. It was like complete biological sciences or life sciences. People are Ashika, your hand is raised. Still, you want to say something? All right, maybe I should start the session now. So, So in this session, today we will just briefly uh, brush up the contents or summarize the content of week one and then look at few of the questions. Okay, so in general, what we will start with is like what is biology, because the course is basics of biology. So we will start with, it will be a very general overview of what biology is and why do we need to study that, and slowly, very slowly, we will move into a little more detail and specifics of classification and diversity in organisms. All right. So I will start sharing my screen. Hopefully my screen is visible to all, right? All right. Um, just all right. Maybe I should switch off the night light. So is it better now? Okay. So there might be some network issue. I will start presenting. Okay. And actually, while I'm presenting, people might still continue joining in. So just let me know in case someone is waiting at the, like waiting to join the meeting. Because while I'm presenting, I won't be able to know if someone is waiting and wanting to join the call. All right. So we'll start. S- All right. So now what is biology? I mean, most of you are from biotech and BSc backgrounds, bioengineering background also. I saw some of them writing about it. So, very briefly, biology is actually the scientific study of complex adaptive living systems. And definitely, as you, most of you, not most of you, actually, all of you might be knowing that living organisms are made up of cells and they can self regulate, reproduce, adapt, and evolve. So, if we talk about the cell, it is the fundamental unit of life, and it can be like it is similar to a microfactory or embedded system where we can actually bring the analogy of the cell taking in inputs via the nutrients or signals. Then the cell processes them using biochemical pathways, and then it gives out outputs like proteins, energy, signals. All right. And biology studies life at multiple scales. Molecular, starting from the very basic molecular level, it moves up to cellular level, tissue, organ, organism, and finally it forms the complete thriving ecosystem. So, living organism is a self-organizing system that processes information, consumes energy, and maintains stability using feedback loops. And in general, if we talk about life, it involves the information flow, usually the genetic code, which is the DNA, then it is transcribed into RNA, and RNA codes for proteins. Okay. Rather, the nucleotides codes for amino acids, which are the building blocks of proteins. Then there's energy flow or metabolic pathways, and then feedback and control mechanisms. So, biology is about understanding and controlling complex systems. So, there are few core branches of biology like cell biology, which basically studies the functions of cells and the metabolism going on therein. Genetics and molecular biology uh, like studies the biological information storage, how, why, and how these things are carried by the cells. These are what is covered under this particular branch broadly. And physiology studies the functional behavior of organs. Microbiology studies microbes, and ecology studies large scale biological systems. And in the context of engineering, uh, one can devise biomedical devices and diagnostics, biotechnology and pharmaceutical applications. The studying biology, different branches of biology, uh, can have significance in biotechnology and pharmaceuticals. Right now, AI inspired by neural systems are also studied very, like, many labs, many researchers are looking into this aspect as well. There are bio inspired robotics as well. Then there's environmental and sustainability solutions as well. So each branch of biology can actually be mapped to an engineering problem. For example, genetics treats DNA as an information system, which is very similar to the data storage and transmission. Physiology studies how organs behave dynamically, and this is very close to the system dynamics and control engineering. And like microbiology is really important, as microbes are used as biological machines to produce insulin, vaccines, enzymes, and biofuels. So this is taken from the lecture slide only, where one looked at the very early biologists. Okay, like Aristotle, he was a Greek philosopher, and he's known as the father of biology. Galen was a Greek physician. He was the first person to perform dissection, also known as father of anatomy. Vesalius was the first person who performed first dissection on humans. Then Malfighi was an Italian physician and anatomist. He is the founder of microscopic anatomy. Then William Harvey, he actually showed how the heart pumps blood and the how blood circulation takes place. Anton von Leeuwenhoek was the first to use microscope and discovered microbes, protozoans, which he termed as animalcules. And then we have Charles Darwin. Uh, like he wrote the book on origin of species by means of natural selection. So if we talk about the history and development of biology, ancient biology, like Aristotle classified animals, Theophrastus or Theophrastus, he was also known as the father of plant biology, father of botany. And mostly ancient biology was observation based science. Now, if we come towards the Middle Ages, the knowledge were preserved in Asian and Arab civilizations. Use of medicinal plants came into picture, and now, right now, like little towards when like all the discoveries were slowly being made and it gained momentum. Microscope invention, Robert Hooke discovered cells, Charles Darwin looked at the evolutionary aspect of organisms, then Watson and Crick gave us DNA model, DNA structure, and present day biology explores genetics, biotechnology, systems biology, data driven, and model based research. So, now if we try to define life in biology, in biological terms, it is actually a very scientifically complex term to define, even though living things can be easily recognized. And biology defines life based on the processes and characteristics, not really only based on the appearance. And a clear definition of life will help us to really classify living organisms and study them, study the origin of life as well, and guide the search for life beyond Earth, like looking at the extraterrestrial forms as well. So, life is best understood as a combination of properties, not really a single feature. Now, when we talk about the core characteristics of living organisms, one is cellular organization. All living things are made of one or more than one cells. Metabolism, there are all the networks and numerous chemical reactions going on that either need energy or give out energy or and produce different kinds of macromolecules. And then homeostasis, regulation of internal conditions like temperature, pH, water balance. Growth and development, increase in size and complexity guided by genes. Reproduction is the transmission of genetic information. response to stimuli, ability to sense and react to environmental changes. And another is evolutionary adaptation. Populations evolve over generations via natural selection. Now, there are some gray areas while defining life, like viruses. The viruses have genetic material, but they do not show independent metabolism. They evolve and reproduce only inside host cells. Then there are prions, which are infectious proteins without any genetic material. Then fire and crystals are there as well, which actually show isolated lifelike traits like growth, some type of organization, but these are not really alive and lack the biological organization. Okay. And why defining life actually matters? Because it improves biological research and classification, it guides medical treatment strategies, it also supports, astrobiology and space exploration, encourages philosophical understanding of life and existence as well. Now, the diversity and complexity in biological system is mainly due to, like, mainly based on the shape of different species varies a lot. Then size of different organisms can be like it can range from a very tiny virus to a giant blue whale. Spatial distributions, living organisms are distributed from extreme cold weather to really hot areas of the planet. Then biological activity, depending on their distribution according to the climatic conditions or the environmental conditions the organisms thrive in, the species actually have to adapt, or actually have different specified or specialized metabolic processes that help them thrive in a particular environmental condition. Now, this is an article written by, like a very short article written by Madhav Gadgil. He was an ecologist, Indian ecologist. Like, we lost him very recently in January this year. So, he suggested like living organisms continually evolve to occupy newer environmental regimes. In the process, they develop more complex structures and grow to larger sizes. They also evolve more intricate ways of relating to each other. The larger, more complex organisms do not replace the simpler, smaller ones. Rather, they come to coexist with them in increasingly complex ecosystems. This actually promotes a continual increase in the diversity of life over evolutionary time. So, if you want to read this article, it's a short article. Like, eventually, the slides and the video will be uploaded in YouTube, like whatever tutorial videos are being taken, and the slides will be shared as well. So if you guys want, you can always, the link is shared, so you can go and read this article as well. So evolution of complexity in living organisms, if we talk about uh, this aspect, so organisms evolve to occupy new environmental niches. They develop larger sizes and complex structures. So complexity increases interaction diversity and leads to coexistence with simpler organisms in ecosystems. So, three major ways of life for energy acquisition: decomposers, which feed on preformed organic matter. Then there are autotrophs, like which actually the organisms who can prepare their own, synthesize their own food, uh, mainly photosynthesis to manufacture organic molecules. Then there are heterotrophs, which feed on other living organisms and also are part of really complex food webs. Then there are variation within these modes as well. Plants adapt to light, moisture, soil conditions. Animals evolve diverse feeding strategies. There are predators, grazers, parasites. Then, based on the size and cellular cooperation, multicellularity evolved to achieve larger sizes and specialized functions. And there are organelles inside the cells like chloroplasts and mitochondria, which actually arose from smaller ancestral cells. All right. Now, when we talk about the interactions, mutualism, and species turnover, in a mutualistic relationship, what happens is it increases the survival and ecological complexity. For example, there are flowers and their pollinators as well. There are fruits and the dispersers of seeds as well. Then there are gut symbionts, which help to digest cellulose or wax in cattle or wax in honeybees. Then there's predator prey coevolution, where predators and prey actually evolved adaptations to outcompete each other. Then there's growing diversity over evolutionary time, like larger complex organisms coexist with smaller simpler ones, and ecosystem complexity actually drives the species packing and geographic turnover. Now when we talk about species turnover, what happens is it environmental gradients like temperature, moisture, or humidity replace species by the ones which are better adapted or better suited to the thriving environmental conditions. For example, like there are different macaque species across India. There are species endemic to like Andaman and Nicobar Islands, which are not really found across the Indian subcontinent. Okay. So why do we really have to classify living organisms? Okay. So it actually organizes the huge biological diversity that we see all around us. So millions of species exist, and classification actually helps grouping these organisms based on their similarities and differences, and hence makes studying and understanding life manageable. Classification also reveals evolutionary relationships, which eventually we can see how species are related through common ancestry. It also helps us to track the evolution of traits or features, whether anatomical or physiological features over time. Predictive value, knowing a group's characteristics can also actually allow us to make predictions about unknown species. For example, suppose certain herb is found suddenly, and medicinal plants in the same family may have similar bioactive compounds. Analysis of its secretions, of the plant's secretions and stuff, can actually help us group them into which um, suppose taxa or species the newly discovered herb belongs to or may belong to. Okay. And definitely how like for classifying organisms into different categories, we need to have standardized names, binomial nomenclature. We will look at it in a little more detail, which can help prevent confusion across languages and regions. So mostly for standardized naming or like standardized naming of species, or in general any living organism, usually one uses like how the existing naming scenario is, like one uses really old languages like Latin, which won't really change over time. So that is what will stay really fixed across a huge period of time, so that it stays constant and doesn't eventually change locally or over the time. And why do we really need to classify? Like how can we really how does classification of organism helps us? Ecology and conservation, it helps it helps us to identify endangered species and prioritize conservation efforts. It helps to track species distribution and ecosystem health. Definitely, it has implications in medicine and biotechnology with classification guided drug discovery from plants and microbes. It helps us to understand pathogens by grouping viruses, bacteria, and fungi. Then agriculture and industry, it enables crop improvement, pest management, and selection of beneficial microbes. And definitely scientific research, which helps organize the different kinds of organisms for experiments, ensuring reproducibility and clarity. Now, how to really classify the organisms? So in earlier days, when number of species were limited, Aristotle actually had used morphological differences as one of the criteria to group the organisms into plants and animals. But eventually, as the diversity went up and the huge number of diverse organisms were there around, so this classification didn't really uh, wasn't really suitable for accommodating a large variety of organisms which don't really fall into plants and animal uh, divisions. So then R.H. Whitaker came into picture, who grouped the organisms using five kingdom classification, and the basis of this five kingdom classification was uh, the classification was based on cellular structure, body organization, what is the mode of nutrition, and reproduction. So the five kingdom classification, the five kingdoms are Monera, Protista, Fungi, Plantae, and Animalia, where monerans are prokaryotic or bacterial cells, which are single-celled organisms. Then there's protista, which are eukaryotic. Fungi eukaryotic, like except monorans, all are eukaryotic. Okay, then cell walls in monorans, it's a non-cellulosic cell wall. In protistans, the cell wall is present. However, the composition may vary across organisms. Fungi, cell wall is present. In plantae, non-cellulosic. Animalia, it's absent. Starts from cell membrane. Nuclear membrane, that is what is the difference between prokaryotes and eukaryotes. Like prokaryotic organisms do not have a well defined nucleus, rather they do have a genetic material, but they have a like without any compartmentalization, they have a very like not really very nicely compartmentalized or membrane covered region with concentrated genetic material. They have a nucleoid as compared to nucleus, which is present in all other organisms like eukaryotes. And another thing is prokaryotic organisms do not have membrane bound organelles, okay, as compared to eukaryotes. And monorans are unicellular, protista are unicellular as well. Fungi, from then on, like from fungi, the organisms are multicellular except one fungi, which is yeast, which is unicellular. Mode of nutrition in monorans can be autotrophic or heterotrophic. In protista, it's autotrophic. In fungi, it's heterotrophic. Plantae, autotrophic. Animalia, mostly heterotrophic. Okay. Now, when we talk about kingdom Monera, it is a group of prokaryotic organisms that are unicellular and lack a nucleus. Okay. Moreover, the genetic material is kind of distributed, where it's known, like in this case, it's known as nucleoid. It doesn't have a defined boundary. And these are the monorans are the oldest organisms on earth, and all living things have developed from them. And kingdom Monera includes two main groups. One is Archaebacteria and Eubacteria. Archaea are the older ones, Eu are comparatively recent ones. So monorans live in extreme conditions of the environment, such as high temperatures, high salt concentrations, hot springs, deep oceans, and salt marshes. And monorans can be either autotrophic or heterotrophic. They are also found in moist environments like snow and deep oceans, as well as they can live as parasites inside other organisms. Okay. So, what are some of the characteristics of monorans? Monorans are single-celled organisms. They have 70s ribosomes in them. All right. So, ribosomes are basically protein um, synthesis factories inside uh, cells of any kind, whether it's a bacterial cell, prokaryotic cell, or eukaryotic cell. So, ribosomes are actually ribonucleoprotein machineries. Like I'm trying to explain the term 70s here. What is there is like ribosomes are actually ribonucleoprotein machineries. It is made up of ribosomal proteins and ribosomal RNA. Okay, now the term S refers to Swedberg constant. Okay, so Swedberg constant actually now in general when we talk about ribosomes, prokaryotic ribosomes and eukaryotic ribosomes. Prokaryotic ribosomes are 70s and it is made up of two subunits, one smaller subunit, one larger subunit. Okay. And the smaller subunit has 30s is called 30s ribosomes, and the larger subunit is called 50s ribosome. Okay. And so what happens here is the Swedberg constant actually refers to the time it uh, takes for centrifugation and for these ribosomal entities to settle at the bottom of the centrifugation tube. Okay. So, so 70s, 70s are the like 50s larger subunit and 30s smaller subunit combined together. So it's not really additive. Okay, so 50s, if separately we are centrifuging it, it will it will like according to the time, it is 50s, and 30s actually will eventually settle comparatively at a lesser amount of time as compared to 50s. Okay, but when 50s and 30s are combined, it's not really additive that you will get an 80s ribosome. Rather, what you will get is when those two are combined together, the time for these two combined subunits to settle via centrifugation, the time it takes, uh, that is 70s. So it's not really that you are adding 50s, 30s, combining, and then you will get 80s. Similarly, for eukaryotes, you have 60s larger subunit and 40s smaller subunit of ribosomes. So 60 plus 40, eventually you don't, when these two subunits assemble for protein synthesis, you don't eventually get 100s ribosomes. Rather, you get 80s ribosomes based on how long it takes for them to centrifuge, okay, and settle down. And so in mononeurons, the nuclear membrane does not enclose the DNA, which is exposed in the form of nucleoid here, this part. Mononeurons are devoid of cellular organelles like lysosomes, plastids, centrioles, Golgi bodies, mitochondria, and so on. And with the help of binary fission or budding, they reproduce asexually. Actually, sexual reproduction like conjugation via pili also takes place, but it is comparatively less prevalent as compared to asexual reproduction. Then there are peptidoglycan layers which make up the cell wall of prokaryotes. And prokaryotic cells have a locomotory organ called the flagellum here, these extensions. And mononeurons or prokaryotic cells exhibit many feeding strategies like autotrophic. They can exist as parasites, they can be heterotrophs, and saprophytic, which feeds on sap. Okay, sap feeding organisms. Now, archaebacteria. This bacteria belongs to like this bacterial group has distinct cell wall structure to allow their survival in extreme conditions. And these species live in harsh habitats like extremely salty areas like halophiles, hot springs, thermoacidophiles, and marshy areas, methanogens. Um, then there's eubacteria. These are the true bacteria or more recent, like more recent group towards like more recently evolved bacterial organism as compared to archaebacteria. It is they have a rigid cell wall and a well-defined flagellum for motility, and several species belonging to this group contain chlorophyll and photosynthetic, and these are actually photosynthetic autotrophs. And few of the chemosynthetic autotrophs oxidize various inorganic substances like nitrites, nitrates, and ammonia to generate ATP. And this particular um, reaction to produce nitrates, and ammonia, and this generation of ATP is actually exploited in industrial settings and as well as in the environment to recycle nutrients like nitrogen, phosphorus, iron, and sulfur, which are actually required by plants. Next, coming to kingdom Protista. Protists are actually a diverse group of mainly unicellular eukaryotes, and they do not fit neatly into plants, animals, or fungi. Their cell structure is eukaryotic. They have a nucleus and membrane-bound organelles, mostly unicellular, and some actually exist as colonies and or simple multicellular forms. Their habitats are mostly freshwater, marine, and moist terrestrial environments. So again, a few groups under this particular kingdom are chrysophytes, which are composed of diatoms and golden brown algae. These organisms are present in water and float passively. Diatoms basically have silica wall and they accumulate in the ocean to give rise to diatomaceous earth. And this particular soil is actually used in the polishing industry, filtration of oils, as well as in syrups. Then there's dinoflagellates, which are photosynthetic marine organisms having different colors depending upon what kind of pigment is actually present in their cell walls. The dinoflagellate cell wall has cellulose on its outer surface, and most of the dinoflagellates have two flagella attached to their cell body. Red dinoflagellates, upon multiplication in the ocean, gives rise to red color. Then there's another section or another group which are euglenoids. which are photosynthetic, and they actually perform photosynthesis in the presence of sunlight. But again, when sunlight is not there, it actually can catch tiny preys and behave like heterotrophs. And the cell body of euglenoids have cell bodies have a rich protein layer called pellicle, which eventually helps the organism to move, giving flexibility to its cell bodies. Then there are slime molds, which are saprophytic protists. They feed on organic matter from decaying twigs and leaves. And during unfavorable conditions, they actually form spores which are resistant and can survive in extreme conditions. And actually, the spores of these organisms can survive for years and are actually dispersed via air. Okay. Then we have Plantae kingdom, which are again subdivided into algae, bryophytes like mosses, ferns, conifers, flowers, like flowering plants, not flowers, which are angiosperms. And mostly, if we talk about the characteristics, it's eukaryotic, having a well defined nucleus and membrane bound organelles. It's autotrophic, where it uses sunlight to synthesize um, organic compounds. Plants have cell walls containing cellulose, and it can reproduce via alternation of generations, either as haplontic, or like N, having an N ploidy, or diplontic, or having two N ploidy. Okay. So if we talk about cellulose, it is actually a polysaccharide. It is one of the most abundant organic compounds in the biosphere. And actually, if we look at cellulose, it is an unbranched polymer of glucose, which are joined by these two, like if it beta 1,4 glycosidic linkage, beta 1 from the first molecule, um, and the fourth carbon from the second molecule are actually joined via dehydration, where H2 molecule is given off and this glycosidic linkage is formed. And it actually, this type of linkage helps in forming really long straight chains. Okay. Fibrils are formed by parallel straight chains that interact with one another by hydrogen bonding. Now, one chain is there, so another chain will be there like this. One chain is there, another chain will be there. So these are like polymers, polysaccharides here, and then there are hanging oxygen and hydrogen atoms, right, in the side chains. So those via hydrogen bonding, they eventually form um, like parallel straight chains and form the cell wall. Okay. And photosynthesis, this is a sugar producing chemical reaction which occurs in chloroplasts, one of the organelles in plant cells. And this is the reaction. And this reaction actually takes place inside thylakoid, which are actually membranous structures. These are the ones uh, wherein the this is a chloroplast. Okay. And these are the structures or membrane bound structures wherein this kind of reaction takes place. Now, if we talk about evolution of plants, it first started as non-vascular, like mosses, hornworts, liverworts, which are ancestral green alga. Then eventually the vascular system in plants developed with non-seeded plants like ferns, and then there were seeded plants like and then flowering plants, gymnosperms and angiosperms. Angiosperms are the flowering plants. Okay. So if we talk about flowering plants, they have roots, stems and leaves. They reproduce by seeds. They produce seed-bearing fruits. And non-flowering plants, mosses and liverworts, they do not have any true root, stem or leaves. They reproduce by spores, and spores are found in capsules like red moss, Leucobrium glauum, Diphysicium foliosum. Then there are ferns, which are the first vascular plants. They have roots, stems, and leaves. They reproduce by spores, and spores are found on the underside of the leaves. You will see if you have a fern plant and you kind of put it upside down, you will see spores, very rough, patchy appearance with the spores. You can see very nicely visible, distinct spores on the backside of the fern leaf. Then there are gymnosperms, which have again roots, stems, and leaves. They reproduce by seeds, but they do not flower. And seeds are not found in fruits. Rather, you might have seen conifers or the cones, and like pine, white spruce, etc. Now, kingdom Animalia. There are invertebrates and vertebrates. If we very broadly divide kingdom Animalia, invertebrates without any backbone, with backbone. Without any backbone, again, it can be divided into without joint legs or with joint legs. And with backbone, one can divide it into fish, reptiles, birds, amphibians, and mammals. We humans belong to mammals. So if we talk about invertebrates, animals without jointed legs, there are segmented worms where the bodies are divided into segments or parts and are found in a variety of environments. For example, earthworms, leeches. Then there are mollusks, which are shelled animals. They have a soft body and may be covered by a shell. They use muscular movement, for example, snails, octopuses. Then there are spiny skinned animals, basically echinoderms, and the bodies are radially symmetrical with five-part body design and found only in oceans, move with tube feet. And examples are starfish, basket stars. Now, if we talk about animals without jointed legs, again we have sponges, porifera, large number of pores present on their bodies. Then there are stinging cell animals like jellyfishes, ctenophores, and sea animals with tentacles and stinging cells. They also have radial symmetry. Flatworms, they have flat ribbon-like bodies which usually thrive as parasites. For example, flukes, tapeworms. Then there are roundworms as well, have long cylindrical bodies with pointed ends. Many are parasites like pinworms. Now, if we talk about jointed legs, arthropods, again it can be divided as with three pairs of legs like insects, with four pairs of legs, arachnids, with more than four pairs of legs, millipedes and centipedes. So bodies are divided into segments or parts. Bodies are covered with an exoskeleton, which is a hard substance that keeps the shape of the animals, maintains the shape of the animals. For example, butterflies, beetles, termites with three pairs of legs, four pairs of legs, spiders, scorpions, mites, ticks, more than four pairs of legs, millipedes, centipedes, prawns, crabs, etc. Now, if we talk about vertebrates, fish, amphibians, reptiles, birds, and mammals. Fishes are cold-blooded. Bodies are often covered by scales. They breathe using gills, can live in fresh water or seawater. Then we have amphibians, which are cold-blooded. Bodies are covered by moist skin without any scale, and they breathe through lungs. They live partly on land and in water, like toads, frogs, etc. There are reptiles, which are cold-blooded. Bodies are covered with dry skin and usually have horny scales. They breathe through lungs and mostly live on land, like lizards, crocodiles, tortoises. Then there are birds or aves, which are warm-blooded. They have beaks and feathers. They have wings, but not all birds can fly. They breathe through lungs. They lay eggs with shells. Ducks, chickens, penguins. Then finally comes mammalian group. So mammals are warm-blooded. Bodies are covered with hair. They breathe through lungs, and mostly, uh, not all of them, but most of them give birth to young ones. Okay, humans, cats, chimpanzees, dogs, etc. Now, out of there are many other organisms which do not really fall under this category. So, like viruses, these are non-nucleated and acellular organisms, and virus can be stored for for years as particle, and they are considered to be false living organisms because they depend on host organism to provide cellular machinery for their own replication or multiplication, and they contain genetic material either as DNA or RNA, and they usually have protein coats as well. And after infecting the host organism, they integrate their genetic material into the host genome to control the cellular machinery of the host. So the host cell can be either a bacterial cell, plant cell, or animal cells. And mostly they are pathogenic and are responsible for different kinds of diseases in bacterial, plants, and animals. And there are viroids, which were discovered by Diner, and these contain only free RNA, and they're actually smaller than viruses, and these viroids do not contain protein coat. Then there are lichens, actually an association of algae and fungi. In this association, algae is the phycobiont and it is responsible for preparing food, whereas fungi is responsible for the fungus, associated fungus is responsible for absorbing mineral nutrients and water. So this association is actually a symbiotic association where both the participating groups are actually helping each other. And actually lichens are also called as pollution indicators because uh, in polluted areas they do not grow in like really polluted areas. And finally there are taxonomic categories, like we looked at the different kingdoms, right? Monorans, protista, uh, fungi, plantae, animalia. Now each kingdom is a group of similar organism, but again these organisms are further divided into different taxonomic categories, like phylum, where different organisms with similar properties are placed. Then class, different classes are present within a single phylum. Then there's order. Now each class is divided into different orders based on aggregates of characters. Then comes family. Individual order is divided into different families. And then genus comprises of similar species with common features. And finally, species, which is the smallest unit in the taxonomic classification. Okay. Now, when we very briefly touched about nomenclature of living organism, so taxonomic classification is used to name an organism. The most popular method is the binomial nomenclature system, which actually uh, comprises of the genus and species info to name each organism. And this system was actually proposed by Carolus Linnaeus, where genus and species are the first and the second words in the scientific name of the living organism. For example, we humans are named as Homo sapiens, where Homo is the genus and the sapiens refers to the species. And in case two organisms belong to the same species, but they may again be subdivided in like according to different subspecies. Okay, so in that case, the subspecies can also be included. And many times the name of the author appears at the end of the biological name. For example, Mangifera indica, Lin indicates that mango was first discovered by Linnaeus. Okay, so Mangifera here is the genus, indica is um, the species, and it is the scientific name of mango. Now, if we look at the classification of kingdom Animalia based on common features, first is levels of organization, cellular level of organization or tissue or organ system level of organization. So if we look at the levels of organization, cellular level of organization, we have porifera, the sponges, and again tissue, organ and organ system level of organization. We can divide this particular all the organisms falling under this. Sorry, Geetika, can you please mute yourself? All right. So all the organisms that fall under that actually have tissue or organ system level of organization can again be divided on the basis of the symmetry that their bodies possess. Okay, like radial and bilateral symmetry. So the radial ones are actually divided into Coelonterata and Ctenophora, And bilateral symmetry, radial symmetry as in you can actually draw a circle, and accordingly along the radius of that particular circle you can actually get two equal halves of the organism along the radius. And bilateral symmetry, the body of the organism can actually be divided into equal left or right halves, or upper or lower halves. Okay. um, Bilateral symmetry, again, the organisms which show bilateral symmetry, they can actually be further divided into uh, based on their body cavities. Without body cavity, acoelomates. Coelom, body cavity is also referred to as coelom. Okay, so without body cavity, acoelomates. With the false coelom, those are known as pseudocoelomates, and with the true coelom like us, um, we are coelomates. Okay, so we will eventually go through this briefly. Now, so the grade of organization, animals are grouped as unicellular or multicellular based on the number of cells. Then symmetry, we looked at it is a plane of arrangement of body parts. Radial symmetry and bilateral symmetry are the two types of symmetry here. In radial symmetry, the body parts are arranged around the central axis. Okay. If we cut through the central axis in any direction, it can be divided into similar halves. For example, hydra, jellyfish, starfish. And in bilateral symmetry, the body parts are arranged along a central axis. If we cut through the central axis, we get two identical halves. Okay. Then we have germ layers. Germ layers are formed during the development of an embryo. These layers give rise to different organs eventually when the embryo becomes an adult. Now, if an organism has two germ layers, the ectoderm, which are the ectoderm, the outermost one, and the endoderm, the innermost one, the organism is said to be diploblastic. But if the organism has three germ layers, the ectoderm, outermost one, the middle one is the mesoderm, and the endoderm, the innermost one, these are such type of organism is called a triploblastic organism. So diploblastic and triploblastic organization, the ones that we talked about in the previous slide. Ectoderm is the outermost one. Then there's endoderm, and there's an undifferentiated mesoglial part, middle area. Okay, so in diploblastic animals, this outer ectoderm and inner endoderm is separated by this mesoglial region, and in triploblastic layer, outer ectoderm is there, very nicely differentiated mesoderm is there, middle portion, and endoderm, the inner layer, is nicely differentiated. So triploblastic organisms, we have it from platyhelminthes onwards, like tapeworms onwards, and jellyfish, sponges. We have these are all diploblastic organisms. Now we will talk about the body cavity. Coelom refers to a fluid-filled cavity inside the body. It separates the digestive tract and other organs from the body wall. Now, when we talk about a coelom or a true body cavity, it is the one that is located within the mesodermal layer here. Okay, and mesodermal layer here. And organisms like the earthworm are coelomates or eucoelomates because they have a true coelom. Tapeworm is an example of an acoelomate because it does not have a body cavity here. If you see, this is the outer layer, this is the middle layer, and this is the endoderm, which is an undifferentiated mesoderm, and endoderm is there. However, in case of a pseudocoelomate, you see the ectoderm is lined by this mesodermal layer, and the endoderm is um, actually separated. However, it's not really lined by any other layer here. Whereas if you see in a true coelomate, the ectoderm is lined by mesodermal layer, and this endoderm is also lined by a mesodermal layer, and the cavity formed therein represents, actually holds different types of or different kinds of organs for of that particular organism. Okay, so pseudocoelomates, animals like roundworms, have a body cavity, but it is located between the endoderm and the mesoderm, and this is considered to be a false coelom, and these organisms are pseudocoelomates. This one here, B. Next, another thing is having a notochord. Notochord is actually a rod-like structure made up of tightly packed vacuolated cells which actually runs along the mid-dorsal line. Animals showing a vertebral column are called as vertebrates, and animals without a vertebral column are invertebrates. Animals are classified into chordates and non-chordates based on the presence or absence of a notochord. Now we quickly will rush through kind of the different phyla like Porifera. So poriferans are aquatic, mostly marine, except one family, Spongilidae, which lives in fresh water, and they are sessile. That means they are actually attached to their substratum, and sedentary and grow like plants. These are multicellular organisms with cellular level of body organization without any distinct tissue or organ. These are diploblastic, they have contractile vacuoles present in some of the freshwater forms, and they actually show a high power of regeneration. And these are few of the examples. Then we have Coelonterata, which can have colonial or solitary forms. Uh, they can actually be free living or sedentary, they are actually mostly marine, but some freshwater ones like hydra are also found. Body is radially symmetrical, they show tissue level of body organization, and body cavity is actually called as coelonteron, which actually helps in digestion and circulation. There are two body types, polyps and medusa. Polyps are actually sedentary and cylindrical life forms, whereas medusa are actually free floating, free swimming, and umbrella shaped life forms. And the mouth, these are the tentacles here. The mouth is surrounded by tentacles which actually help in locomotion as well as capturing the prey. And there are cnidocytes, which are specialized cells containing a stinging structure called nematocytes. And these nematocytes help the animal to catch the prey because nematocytes can discharge toxin onto the prey organisms. They have a poorly developed nervous system. Reproduction can be both sexual or asexual. Hydra actually reproduces asexually by budding, and um, and the sexual reproduction involves the gametic reproduction. Okay. Now, when we talk about Ctenophora, these are radially symmetrical organisms, mostly marine, having a diploblastic two-layered body organization. These animals are also called as sea walnuts or comb jellies. They have a spherical body with an external row of cilia. Actually, they show bioluminescence as well. They are bisexual animals, and uh, they reproduce by sexual reproduction, showing external fertilization where the gametes are released outside and fused outside the body. Okay. Then we have the flatworms, Platyhelminthes. They are bilaterally symmetrical with blind sac body plan. They are triploblastic but without any body cavity. And mostly these are endoparasites. Body is like a ribbon, unsegmented, dorsoventrally flattened, and covered with a cuticle. The parasitic forms actually also have suckers. Here, suckers or hooks for attachment to the host, and there is no digestive system in the parasitic forms, and excretory system is made up of flame cells. Then we have roundworms, which are Astelminthes, mostly parasitic, and body is long and cylindrical, showing bilateral symmetry. They are again triploblastic but pseudocoelomate, where both the ectoderm and the endoderm are not really covered by the mesodermal layer. And body wall shows longitudinal muscles but no circular muscles. Excretion takes place by protonephridia, and their average lifespan is of 4 months and have a lifespan ranging from as early as 14 days to as long as 80 days depending on the type of roundworm we are talking about. Then we have annelids, which are mostly aquatic, marine or freshwater. Some can be terrestrial uh, or free living. Some can be commensal or parasitic. Body is actually elongated. triploplastic with a bilateral symmetry. They are they possess proper body cavity. They are coelomate, and mainly the name Annelida comes from their ring-like segmentation in the body. Okay, and which is called metameric segmentation. And body organization is of organ grade, and the body wall is contractile or dermomuscular, consisting of outer muscle fiber, circular, and inner longitudinal muscle fibers. Appendages are jointed if present, and locomotory organs are segmentally repeated chitinous bristles called setae or chytae embedded in the skin. And examples are earthworm, leeches, canaries. Okay. Then there's arthropoda or insects, which is the largest and the most successful phylum. They can be solitary or colonial, mostly free living, and they are present everywhere, and the body shows bilateral symmetry. Body is covered by a tough chitinous cuticle, and the body can be actually segmented into three parts: head, thorax, and abdomen. And in some organisms, the head and thorax can fuse to form a cephalothorax. Cephaly refers to head. And arthropods possess legs for crawling, creeping, walking, and wings for flying. Okay. And the organisms show sexual dimorphism. Animals are oviparous, egg laying, and fertilization is internal. And development can be direct or indirect, showing metamorphosis. And in honeybees, individuals are produced by parthenogenesis without fertilization. And definitely, there are economically important arthropods, honeybees, silkworms, prawns, crabs, lobsters. And then there are harmful ones as well, which are vectors for different diseases like mosquito, spiders, cockroaches, centipede, etc. Then we have mollusca, phylum mollusca, where um, the sexes are usually separate, but some can be hermaphrodites. Fertilization can be external or internal. Development is direct or showing metamorphosis through a stage called veliger larva. Head can be distinct, bearing mouth, eyes, tentacles, and other sense organs. The ventral body is modified into a muscular plow, this part. The foot, which is variously modified for creeping, burrowing, and swimming. There can be mantle or pallium, which is a fold of the body wall that leaves between itself the main cavity, mantle cavity. And then there's the visceral mass, which contains the vital organs of the organism in the compact form, taking the form of a dorsal hump or dome. And body cavity is a hemocoel. The coelom is actually reduced and represented mainly by the pericardial cavity, gonadal cavity, and nephridia. And the digestive tract for mollusks is actually simple, with an anterior mouth here and a posterior anus here. But in gastropods, scaphopods, these are further subgroups of phylum mollusca. And cephalopods, the intestine becomes U-shaped, bringing anus to the anterior part. Okay, like it kind of curls, and this part actually comes towards this anterior region. Okay. Then we have echinoderms or starfish, which are spiny skinned animals, starfishes, etc. Exclusively marine, can be solitary, sedentary, or free living, and they show radial symmetry. It will be radial symmetry and pentamerous symmetry. Bodies can be spherical, elongated, or star shaped. Body actually doesn't have a well defined head. And one thing to be noted is the presence of water vascular system in the body for locomotion via tube feet, and water enters the water vascular system through an opening called as madreporite. Examples again are urchins, starfishes, cucumbers, etc. Actually, this water vascular system, if you see, this is the one. It is composed of canals and connecting numerous tube feet. These ones, these small racket-like structures. Echinoderms move by alternately contracting muscles that force water into the tube feet, causing them to really extend and push against the ground, then relaxing to allow the feet to retract. and they are carnivorous and mainly feed on mollusks. They move with their arms and tube feet. Respiration is by peristomial gills. Circulatory system is greatly reduced. Heart is absent. It is of open type. Nervous system is very simple with the ring around the mouth and radial nerve in the arms. Sexes are actually separate. Fertilization is external and development is indirect. And again, these echinoderms also show a high power of regeneration. Then we have hemichordates, which are exclusively marine, usually living at the bottom of the sea. Body is actually soft, fragile, and unsegmented, uh, but it can be divided into three parts with proboscis, collar here, and a trunk. Okay. Buccal cavity gives rise to a rod-like structure, which is considered as notochord by some scientists. Okay, and they feed on microbes present in water. And the proboscis here helps to make burrows while the entire body brings about the movement. And there's the alimentary canal, which is complete, straight or U-shaped. Respiration occurs by paired gills. There are gill slits as well, and which like via which the gills open out. Circulatory system is simple and of closed type. Blood is actually colorless. Nervous system is actually embedded in the epidermis on both dorsal and ventral sides. Sexes are actually separate. Fertilization again is external. Developmental stages are indirect via a free swimming larva existing in between. And this particular phylum is actually considered a connecting link between chordates and non-chordates. Okay. So now let's look at few of the questions. Sorry. So the father of microscopy is, can anyone answer? What will be the correct answer out of these? We also looked at in one of the slides earlier. Yes, the correct answer is option A, Anton von Leeuwenhoek. Now, the ribosomal RNA. So we talked about 70S ribosomes and 80S ribosomes. Okay. Uh, and ribosomes are ribonucleoprotein machinery. That means it is composed of ribosomal RNA and ribosomal proteins. Okay. So now the composition of prokaryotic and eukaryotic ribosomes differ a little. Okay. So it is asking what is the common type of ribosomal RNA present here. Does anyone know? Any idea? So ribosomal RNA, if we talk about prokaryotic ribosomes, I was talking about the larger 50S subunit and smaller 30S subunit. Okay. Now, when we talk about the 50S subunit, it is again made up of 23S ribosomal RNA plus 5S ribosomal RNA. So this 23S ribosomal RNA is actually 2.9 nucleotides, 2900 nucleotides long, and 5S ribosomal RNA is approximately 120 nucleotides long. And then there's the smaller subunit, which is the 30S subunit. It is made up of 16S ribosomal RNA. All these values, the 16, 23, again refers to the time taken for centrifugation. Okay. So 16S ribosomal RNA is actually approximately 1500 nucleotides long. Okay. So when we talk about eukaryotic ribosomes, the complete complex is 80S, where it is made up of 60S larger subunit, 40S smaller subunit. 60S is again actually made up of 28S ribosomal RNA, 5.8S ribosomal RNA, and 5S ribosomal RNA. 40S subunit is made up of 18S ribosomal RNA. Okay. So the question is actually asking us which type of ribosomal RNA is present in both the prokaryotic ribosome as well as the eukaryotic ribosome. So if we see here, the common thing here is the 5S ribosomal RNA. Okay. So the correct option will be option D, 5S ribosomal RNA. Next, the type of association where two organisms function as a single unit and help each other for survival is, Can anyone tell the answer? Yes. The correct answer is option A, symbiosis. Okay. Um, Next, we have the smallest unit in the taxonomic classification with fundamental similarities is, what will be the correct answer? Yes, D, species. Now, the phylum that is closely related to both chordates and non-chordates and is actually considered a connecting link between them. We just, a few slides back only we read about it. What will be the answer? Option D, hemichordates, right? And we talk, so the protein rich layer in the cell body of euglenoids that actually gives flexibility to the organism is, any idea? Yes, good. Option B, pellicle. Now the organism, oh, I have shown the answer then and there. The organism which lives in extremely salty areas is option A, halobacterium. Okay. So the correct answer is option A, halobacterium. Halo refers to salt. It lives in highly saline environments like salt lakes, salt pans. Then there's thermoplasma. Thermo means heat. Plasma, acidic environment, lives in hot acidic environments. Then there's pyrococcus. Pyro refers to fire, like extremely high temperatures. It is a hyperthermophile. And then we have sulfolobus, which lives in acidic hot sulfur springs. Okay. Now, the book that explains how species evolved over time and gradually diverged from each other via natural selection is, this was written by Charles Darwin. The correct answer is option B, Origin of Species. So, origin of life is basically the concept which discusses how life began on Earth, not how, like evolution by natural selection. It doesn't talk about evolution by natural selection. It talks about how life actually began on Earth. Origin of Species, which the question asks for, was written by Charles Darwin, which explains evolution of species through natural selection. The Origins of Virtue is actually a book written by Matt Ridley, which is about evolution of morality and human behavior, not really the evolution of species through natural selection. And there's The Selfish Gene, which is comparatively a very famous book by Richard Dawkins, which actually focuses on the gene centric view of evolution, not the original natural selection theory. Okay, so the correct answer will be for this particular question, Origin of Species, written by Charles Darwin, which looks at how species have evolved over time via natural selection. Now, the radial symmetry is found in which of the following phylum? The correct option is, we're talking about radial symmetry. So it is Cnidaria. Okay. Uh, Jellyfish, hydra, corals, all these will be having radial symmetry, whereas porifera, the sponges, are asymmetrical. Platyhelminthes, the flatworms, show bilateral symmetry. Annelids, segmented worms, again show bilateral symmetry. So the correct answer will be option C, Cnidaria. Now, compound eyes are present in which of the following organisms? Any idea? Yes. Option B, mosquitoes. So, octopuses have actually simple camera type lenses like humans. Mosquitoes have compound eyes. Cockroaches have compound eyes made up of many ommatidia. Frogs have simple eyes with lens. Humans again have simple camera type eyes. So now if we just maybe characterize it in a, like tabulate it, simple eye versus compound eye. So the structure, simple eye has single lens which focuses light on the retina. Compound eyes are made up of many small units called ommatidia, each having its own lens. If we talk about the image formation, simple eye forms a single focused image, compound eye forms a mosaic image, which are actually multiple small images combined. If we talk about light detection, simple eye can detect fine details and color, and compound eyes are excellent at detecting movement but poor at detecting the finer details. Again, simple eyes are found in humans, frogs, octopuses, cats, birds, etc. Compound eyes are found in insects, uh, crustaceans, crabs, dragonfly, mosquitoes, houseflies, cockroaches, etc. Simple eye, this particular adaptation is suited for precise vision, for perceiving the depth, and compound eyes are suited for detecting rapid movement and wide field of view. Okay. Again, I mean, found in and examples are the same. Now, the correct characteristic feature of heart and blood circulation present in mammalia is, can anyone answer? Like it's being talked about us humans, like we belong to the group mammalia. So the correct answer will be a complete four-chambered heart and double circulation. Okay. So if we talk about fishes, chambers, there are two, one atrium, one ventricle, single circulation. If we talk about amphibians, they possess three-chambered heart, two atria, one ventricle. It's an incomplete double circulation. Then there are reptiles. Mostly reptiles have three-chambered heart, um, two atria and one ventricle. Then comes birds and mammals. They have four chambers, two atria, two ventricles, like the upper chambers and lower chambers. And the double circulation is a complete double circulation with a well like distinct separation between oxygenated blood flow and deoxygenated blood flow. Okay. So that's it for today's session. Maybe I will stop sharing my screen. Thank you all for joining. We will meet again in the next, like on next Sunday with week two's content and looking at week two's few of the questions related to week two content. Okay. All right. Acha, Meenu says, can you slow down the lesson a little? Actually, yes, but I couldn't see your chat. You can unmute yourself in between the lesson. Just maybe sorry to interrupt or something. Uh, just unmute yourselves and tell that because I can't see your chats. Okay, so you can just unmute yourself and say. Okay, thank you, Kirtana, for joining. Thank you, Maria. Yes, Neha, you want to say something? Mm-hmm. Oh, okay, 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 okay. So, or maybe I should share the screen again. Okay. Just, Acha, it says nervous system is embedded in epidermis, the outer layer, on both dorsal and ventral sides. Okay, so it has like particular some kind of cells that are there which can help in, like it's a very simple thing, not like us humans. Okay, so basically the dorsal side, it has few linings which help in communicating the some kind of signal from dorsal side to the ventral side of the, like dorsal side is this part, this part here, and ventral side is this part here. Okay, so it's embedded in the epidermis, as in the very outer layer of the, if we talk about, if we tell it as a skin here, then the cells are lined here, kind of. Okay. So you see dorsal nerve cord is there, and this particular thing also helps in relaying signals from its dorsal side to the ventral side as well. Okay, but it's at the epidermis, as in towards the outer layer of the um, skin, if I can say like very vaguely. So this is the thing that is being talked about. Okay. Exact origin, I'm not sure about the exact origin. Um, Maybe I can go back and look at it. Exact origin, which is the, like, if you're talking about, what I can talk about is the ectodermal or mesodermal or endodermal layer. Um, that I have to go back and check. But like the origin of layer can be, what we can what will be the correct way of saying is, from either from the ectodermal layer, endodermal layer, or mesodermal layer, like that we can say something like that. But which exact layer it is originating from, the nervous system, I have to go back and check once, and I will get back to you. Okay. Yes, 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 most probably, but still I will, like, just to make sure, I will just go back and check. Sure, sure. Sure, sure, sure. Yeah, no problem. Thank you for the question. Okay. I will stop sharing. Okay then. Thank you all for joining, and I will get back to you, Neha. Okay. The next session, I will get back to you. All right. Thank you for joining. Then I will end the meeting for everyone now. See you on the next Sunday. Thank you so much.